Hello, my name is Solah and I'm a senior from Jenison High School in Michigan. And this presentation will discuss the research in comparing Turner syndrome and Fragile X premutation carriers, focusing on the convergence to similar pathophysiological outcome. To begin, premature ovarian insufficiency, or POI, is a condition of early menopause from ovarian dysfunction. While natural menopause can occur after the age of 40, POI is an abnormal loss of ovarian function before that age has been reached. It decreases the number of follicle cells in the ovaries, which causes ovulation to cease and produce less hormones. Specifically, POI prevents the development of primordial and primary follicles, which are considered the earliest ovarian follicles. The exact cause of POI is difficult to pinpoint, as it is often caused by various different factors, ranging from genetics to environmental factors. Among these, genetic disorder leading to abnormalities of the X chromosome is one of the most common factors. While there's no known cure for POI, there are several different treatments, such as hormone replacement therapy or in vitro fertilization. In this research, we will focus on two X-linked conditions that will lead to ovarian insufficiencies. The first condition is Turner syndrome. Turner syndrome is one of the most common X chromosome aneuploidy disorder, resulting from a complete or partial loss of the second sex chromosome, or more commonly called the 45X karyotype. Ovarian dysfunction is a common feature observed in women with Turner syndromes. This is an image at the bottom of ovarian morphology in tissues from females with Turner syndrome from research from Elsevier. In the first image, you can see that the primordial follicles are missing the granula or vesicle membranes. And in the second image, the primordial follicles are collapsed or completely empty. The abnormal X chromosome disrupts the normal ovarian functions and the follicle developments, which accelerates loss in X cells as the fetal develops, leading to POI. Fragile X syndrome is an intellectual disability resulting from a mutation in the X chromosome where it expands with the CGG repeat stretch at the FMR1 gene, or the Fragile X mental retardation 1. In this research, rather than the patients with Fragile X syndrome, we will focus on the premutation carriers who showed symptoms of POI. Fragile X premutation carriers, or FXPC, has a partial mutation with around 55 to 200 CGG repeats at FMR1. This can be inherited to a child to a full mutation with over 200 repeats and cause Fragile X syndrome. Carriers often show condition of premutation, or more commonly called the Fragile X primary ovarian insufficiency, FXPOI. The premutation impairs the mitosis of the follicle cells, leading to a degeneration of the developing follicles and ultimately lead to ovarian insufficiency. Now, both conditions have a distinct X-linked genetic disruption but presents an overlapping outcome in ovarian insufficiency. This research will investigate a possible convergence point in the downstream biological pathway of Turner syndrome and FXPC despite having different genetic causes. We will investigate the genetic components of these conditions and cross-reference them to find similarities that could be the possible reason leading to POI. This research uses available gene expression data from the GEO database. Two different databases, one for each condition, was analyzed to identify their genetic makeup using GEO2R and compare the samples based on their gene expression profiles of the top 500 genes. From the genes given, we found a specific gene that overlaps in both conditions and viewed closer using gene ontology. StringDB was also used to explore the gene's role in the condition's biological processes to detect a potential downstream effect from the gene related to ovarian functions. And finally, the information gathered was further reviewed in comparison with other published works to produce a final analysis. And I will explain briefly on how to use each of the tools throughout the presentation. This study analyzed two datasets published by GEO, or Gene Expression Omnibus. GEO is a global public database archive that stores gene expression datasets and gene expression profiles. The select database in this experiment was analyzed by microarray. Dataset GSC 46687 compared the gene expression in Turner syndrome patients, specifically their peripheral blood monocular cells, with 16 maternal X, 10 paternal X, and 10 controls. 45XM indicates maternal inherited X chromosome, while 45XP indicates paternal inherited chromosomes. Although these samples were blood-based rather than ovarian tissues, They provide a valuable insight into gene expression's alteration caused by the X chromosome loss. Now, GEO2R is a web tool that allows users to compare two or more groups of samples from GEO to identify specific genes that are differentially expressed across experimental conditions. The dataset from Turner syndrome was divided into three different groups by 45XP, 45XM, and control. GEO2R identified the top 500 significant genes with a p-value of less than 0.001 and with the largest log-fold change values. This represents the greatest gene expression differences between the control and the samples. The UMAP gives a visualization of the difference in gene expression profiles. The plot combined the 45XP and the 45XM samples to show a difference as a whole, and as you can see, there's a very distinct line in the expression values between the control samples and the Turner syndrome samples. The separate cluster confirms that the patients have a distinct gene expression pattern. Similar steps were taken with FXPC. Datasets GSC48873 contain data from female Fragile X premutation carriers with FXPOI and matched controls from peripheral blood monocular cells, with 5 control samples and 9 Fragile X premutation carriers. Similar to the Turner syndrome dataset, this was also blood-based samples, but still indicates alterations caused by X chromosome loss and allowed examination of transcriptional changes associated with ovarian insufficiency in context of Fragile X premutation. Analyzing the UMAP through GEO2R, FXPC samples also demonstrate a clear separation from the control, confirming a distinct transcriptional profile. The separate cluster confirms that the patients have a distinct gene expression pattern. Although no single gene was common between the datasets among the significant genes selected, two highly homologous ribosomal protein genes were selected, RPL37 and RPL37A. Gene Ontology is an online database with computational models of biological systems ranging from the molecular to the organism level. These analyses show that the paralog genes encode 63S ribosomal proteins belonging to L37 and L37A family, which is a crucial component in carrying out protein synthesis in all human cells, including thyroid follicular cells that plays a role in ovarian function. This is a gene expression profile of the ribosomal protein genes in their respective conditions from GEO2R. A negative log-fold change indicates a downregulation in gene expression, and RPL37 was found significantly downregulated in FXPC with a value of negative 0.89. While the dataset for Turner syndrome did not provide the exact values of log-FC for RPL37A, the expression analysis from GEO2R showed the expression levels of the gene, with bars indicating the expression levels. And as you can see, there's an obvious difference between the control and the sampled expression levels that we can confirm that the gene is downregulated. As these genes are downregulated, it suggests that the disturbance in large ribosomal subunit functions could be the potential convergence mechanism for premature ovarian insufficiency. Now, StringDB is a tool that uses genes to connect known protein interactions and connect the relationship to probable biological processes. These are the connections between the genes in both Turner syndrome and FXPC, and the biological process identified in both conditions through overlapping genes is a metabolic process of ribosomal proteins. Now, if we go back to our expression profile table, Turner syndrome showed a moderate enrichment signal value of 0.33 and a low false discovery rate of 0.008. FXPC showed a relatively high signal value of 0.56 with a low false discovery rate of 9.56 times 10 to the negative 7. Enrichment analysis indicates a disruption in translational and large ribosomal subunit activity with statistically significant results in both datasets. Now, despite the distinct X-linked genetic disruptions, these findings suggest that dysregulation of ribosomal and metabolic process are present in both conditions and could represent a shared downstream mechanism contributing to POI. RPL37 and RPL37A are structural ribosomal protein genes that synthesizes protein in the follicular cells, which requires a high level of protein synthesis to secrete thyroid hormones. Now, disruption in these genes could impact the metabolic enzyme production and trigger ribosomal translation stress pathway, potentially altering the follicular cell and leading to POI in both conditions. Although Turner syndrome and FXPC are different in their genetic disruption, the findings highlight how they could converge on similar downstream process in the physiological outcome of ovarian functions. To sum it up, Turner syndrome and FXPC exhibits downregulation of homologous ribosomal protein genes, and this shared downstream disruption may explain the overlapping outcome of ovarian insufficiency. Understanding this convergence provide insight into mechanisms that could be relevant across different X-linked ovarian disorders. An important thing to note is that the analysis relies on blood-based datasets, as the research on ovarian tissue was very limited in the GEO database. However, with the findings, the connection between the two conditions may inform future studies to identify biomarkers and ovarian conditions for specific areas related to ribosomal protein genes for future POI research. It can also be used to implement therapeutic or diagnostic strategies to target ribosomal translational pathway in developing treatments for POI. These are my references used in the research, and thank you very much for your time.